Have you ever wondered what makes you, well, you? What is it that fuels the incredible, mind-boggling diversity of life on our planet? Today, we're going to explore the fundamental process behind all of it, a constant, high-stakes shuffle that's happening inside every living thing. At the end of the day, it all comes down to this one question. We all share the same basic genetic building blocks, right? DNA. So how does nature manage to create such an astonishing range of outcomes from that same blueprint? Well, turns out the answer is in this constant game of chance and strategy that's being played at the cellular level.
I think the best way to picture it is like this. Think of an organism's genes as a deck of cards. Now, every single time a new life is created, that deck gets a thorough shuffle, dealing a completely unique hand. This constant mixing is the engine of variation, the very thing that drives evolution forward.
But, and this is where it gets really interesting, what if some of those cards were a little bit sticky? What if certain cards just had this tendency to always be dealt together, completely defying the shuffle? This is the other side of our story, a force that holds combinations together, totally against the odds. So let's start with the shuffle itself. This is the main event, really. It's the process that generates the incredible diversity we see all around us. And it's the reason why, unless you're an identical twin, no two people are ever exactly alike. The official name for this is genetic recombination. It all happens when reproductive cells are being formed, and it's driven by two key things: crossing over, where chromosomes literally swap pieces of their DNA, and independent assortment, where they just line up in a totally random order. So it's like both swapping some cards with another player and shuffling the whole deck. And the number of new combinations we're talking about, it's not just big, it's astronomical. We can get a rough idea of the scale with a pretty simple formula: 2 to the power of n, where n is just the number of chromosome pairs that an organism has. So, just how staggering are these numbers? Let's take a simple housefly. They've got 6 pairs of chromosomes. Now, using our formula, the number of possible combinations in just one parent's reproductive cells is 64. But when 2 flies mate, you have to multiply those possibilities together. That gives you over 4,000 unique combinations for their offspring. And that's before we even add in all the extra variation from crossing over. It's just incredible. After seeing just how powerful that genetic shuffle is, you'd think everything is just completely random. But, for just about every rule in biology, there's always a fascinating exception. So let's look at the genes that actually resist the shuffle. Yeah, after all that talk about the massive potential for variation, it's kind of surprising to learn that some genes just don't play along. They seem to have a mind of their own, sticking together through the whole process. So why do they stick together? Well, it's all about location, location, location. As you can see in this diagram, when genes, like A and B here, are physically close to each other on the very same chromosome, that shuffling process is way less likely to split them up. They tend to just travel together as a single unit. This whole phenomenon is known as gene linkage, and it's a huge deal. Why? Because it's a major exception to one of the very first rules of genetics we ever learned, Mendel's law of independent assortment. It turns out that genes aren't always inherited independently of one another. Now, the fact that some genes are linked wasn't a dead end for scientists, not at all. In fact, it opened up this brilliant new possibility, the ability to actually map the layout of our chromosomes. Which of course leads to the next logical question. If these genes aren't always assorting independently, how in the world can we figure out their order and location? Well, the answer, pretty cleverly, comes from the very process that sometimes breaks them apart. Scientists create something called a chromosome map. And this is just a brilliant use of data. By studying thousands upon thousands of offspring, they can measure exactly how often those linked genes get separated by that crossing over event we talked about earlier. The principle behind it is beautifully simple. The farther apart 2 genes are on a chromosome, the more physical space there is between them for a crossover to happen and split them up. So if you see a high frequency of crossing over, it means the genes are far apart. A low frequency? That means they're super close neighbors. And this isn't just some abstract theory. Right here, you're looking at the very first chromosome map ever made. It was for the humble fruit fly, all the way back in 1913. By meticulously tracking traits like body color and eye shape, scientists were able to lay out the actual sequence of genes on the chromosome. What an absolutely monumental achievement. So far, we've been looking at shuffling individual genes. But what happens when the shuffle goes wrong on a massive scale? Well, there's this wild anomaly that involves entire sets of chromosomes, a condition that's lethal for us, but it's kind of like a superpower for other forms of life. This is called polyploidy, and we're not just talking about having one extra chromosome here, we're talking about having one or more entire extra sets. So instead of being diploid, or 2N, like us humans, an organism might be triploid, which is 3N, or even have 6 or 8 sets of chromosomes. Okay, so why should you care about this? Well, take a look at your morning cup of coffee. Many of the plants we rely on for food and agriculture are polyploids. This condition, which would be totally fatal in humans, is often a huge advantage for them. You know, it's surprisingly common in the plant world. The wheat we use for bread has 6 sets of chromosomes, and strawberries have a massive 8. This genetic quirk often leads to larger, stronger plants. But while it's a real game changer for agriculture, it's almost always lethal in animals, and that definitely includes us. All right, so after exploring shuffling genes, genes sticking together, and even extra sets of chromosomes, let's bring it all back to our main theme. What we've really seen are two powerful, opposing forces at the very heart of inheritance. This table really puts that central tension side by side. On one hand, you have genetic recombination. It's the great shuffler, constantly creating new combinations to drive variation. Then, on the other hand, you have gene linkage. It's the preserver, holding on to those successful, time-tested combinations. It's this constant battle between creating something new and sticking with what works. And that leaves us with one last fascinating question to think about. It's all a balancing act, isn't it? Life absolutely needs the stability of keeping successful gene combinations together, but it also desperately needs the novelty and adaptability that comes from shuffling the deck. And it's this constant push and pull between preserving the good and trying for something even better that makes genetics and really life itself so incredibly dynamic.